Miney will talk to him or her.
Awesome.
I might say, yes.
Yeah, let's see. Okay. Great. Okay. Welcome, everybody. This is Agential Topics. I'm Charlie Munford. And Agential Topics is a meeting, periodic meeting, made possible in part by Agential Biology Institute, which is founded because we think that there is a missing causal layer in biology, and we're building the infrastructure to study that missing causal layer. And as part of that, we have these discussions to zero in on possible biomechanical explanations for cellular cognition. And today, without too much ado, we have a wonderful guest, Jack Tuszynski, who's done an enormous amount of work on microtubules and thinking about cells as information processing agents. And in keeping with our tradition of doing very little introduction of the guest, I'll hand it over to Jack quite swiftly here for a presentation and discussion to follow. Thank you so much.
Okay, thank you, Charlie. Thank you, everyone, for joining us. Um, yes, I've spent a lot of time thinking and doing research on microtubules and related topics, not just microtubules. And this is one of the things that I want to emphasize, that microtubules are not alive, okay, and they are not conscious, in case you were wondering, but they play an important role. And I'm a physicist. Currently, my association with the Department of Mechanical and Aerospace Engineering in the Politecnico di Torino, Technical University of Turin in northwestern Italy. But I'm here sitting in Edmonton, Canada, where my home is, and I've been a professor of physics and for 15 years of oncology at the University of Alberta, where most of the work was done. Let's see. Okay. I published a little bit of stuff on this topic, also related to consciousness. These three books have some aspects of consciousness discussed from the physics perspective. And so I'm interested in things related to consciousness, but information, biological information, memory, storage of information and retrieval and processing. And that kind of is the hierarchical organization of the structures which, as we know, do this stuff. And we don't know how that happens by and large. I mean, neurophysiology tells us a lot, but according to many, myself included, there's more than meets the eye. So I'm not going to talk about what is consciousness, but these questions, maybe in discussion, we can talk about this. I will say a little bit about where memory can be stored and how much can be stored, and also some interesting topics from the point of view of physicists, the computational power of the brain and the um, energy expenditure to do these things, and compare it to some of our technology. Um, and then information processing and topics, is it classical, is it quantum, maybe hybrid, maybe fractal, something like that. All right, so let's start with some numbers. Um, as you may know, we have about, on average, something like 100 billion neurons in the brain, and we'll peer inside the neuron to see what's in it and how this can be more than just a binary switch, which I think is offensive to the neuron. And then each neuron has about 10,000 synapses. On average, can be much more, can be a bit less. And so if you just multiply these numbers, you get 10 to the 15th synapses. If they operate at, conservatively speaking, 10 impulses per second, you have something like 10. Again, these numbers are maximum, right? 10 to the 16th operations, if these operations are understood as information processing of some kind, 10 to the 16th operations per second, which is enormous. Blue Gene supercomputer that was used to beat Kasparov in chess and further versions of it, let's say operate at 10 to the 16, 10 to the 15th operations per second, but these supercomputers consume as much energy as a small town or mid-sized town, so something like 1.5 megawatts, million watts, so joules per second in physical terms. Compared to the brain, which although it's a high energy consuming organ, about 25% of our metabolic energy is consumed by the brain. So that's 25 watts, which is, and much of it is just to maintain normal functions, temperature and physiology. So if you conservatively assume that 10% of it is for cognition, or maybe 15, whatever, you're talking about a million times, or comparable to the energy consumption by a flashlight. And that's what the brain uses for information processing, in my estimation. Some people maybe consume more. Grandmasters in chess, when they play chess at a high level, I'm sure that they consume a lot more. Or when you perform some demanding creative activities, but this is the order of magnitude. So, and on top of that, of course, we do many other things. So our brains perform not just process information, cognitive, but the orchestration of the entire body's activities. So and last time, I don't know if most of you attended this, Marco Cavaglia and Tommaso Firo talked about the electromagnetic perspective of the brain's function in terms of the so-called hollow brain theory. I will go deeper, and they focused on the membrane. While the neuronal membrane, of course, is super important because of the action potentials which propagate electric signals. In fact, electric field transitions. What's happening inside is, in my opinion, equally important. And I don't want to say one thing is more important than the other, because all of it is important. I think if you were to dissolve the membranes of neurons, nothing will happen. Even if you were to weaken the mechanical strength of the membranes, you will not function as a cognitive person. On the other hand, if you were to provide we know that if microtubules are disturbed in some way, either mechanically or electrically, the same will happen. And one example of it is traumatic brain injury, where studies have shown that microtubules of TBI patients post-mortem were broken. So there is that. And this is kind of where I want to go, going deeper into the structure of the neuron and look at, you can see here an artist rendition. This is from Travis Craddock, my former PhD student, who is now professor at the University of Waterloo. And you can see inside not just microtubules, but mitochondria. Mitochondria move on microtubules with the power generation by motor proteins, and then inside. Oh yeah, we'll go into this. This is a tubulin dimer, which is the building block of microtubules, and then you have the reactive. You have, of course, reactive oxygen species as a byproduct of mitochondria. Some people say that they play an important role in some form of electromagnetic interactions. Let's put it this way for now. Maybe even providing energy for some form of electromagnetic processing. Okay, so that's kind of the scheme of things. And just recently, I found on LinkedIn this gentleman, Joseph Jacks. I think he's a financial analyst or investor. I'm not quite sure. he pointed out that the simple, and this is actually paramecium, a single cell organism, which has fairly, complicated range of functions, but doesn't have a nervous system. It says here about 100,000 microtubules. It may be less. I'm not sure of that many, but let's say 100,000 microtubules, and they reorganize and respond to external stimuli, temperature gradient, chemical gradients, mechanical gradient, and so on. So there is definitely here a kind of a micrometer size living object, which is showing some level of intelligence, to say the least, and without the brain or a neuron. It's just a cell with microtubules and mitochondria, as you can see here, which supply the energy to it. And so the same structures organized in series of brain neurons and other cells. So it's worth trying to understand what they do. And of course, microtubules, in addition to being present densely packing axons and dendrites, they are also present in all the eukaryotic cells and form different morphologies. Here you have on the left hippocampal neurons with tubulin green and actin in red. And then you have of course, super important aspect of cell division. Mitotic spindles are composed of microtubules, and human mitotic spindle, each spindle has about 30 microtubules. You're not seeing slides? Are you guys seeing slides?
I see them.
You probably need to change the
resolution.
The little thing on the top. No, Wayne. Yeah, you should be able to have like a little thing at the top that says meeting and then Jack's screen.
Yeah. I think they'll be good.
So you're probably in the wrong tab.
He might be in the tab that shows people's faces instead of this meeting tab.
The least interesting tab.
I think
it depends on the perspective.
Yeah, I think you're set up right, Jack.
All right. So I'll continue. And okay. So then you see microtubules forming these pathways inside the cell over which mass transport is taking place, including kinesin and dynein moving in opposite directions, the motors. So it's incredible machinery carrying mass movement from place A to place B. All right, so we know all of this. And some more illustrations of what microtubules and how beautifully this is all orchestrated, including neurite growth, brain development, and so on. And importantly, in this context, they also form supramolecular structures, such as cilia and flagella for cell motility in single cell organisms, and also importantly, the centriole, the centrosome, which is composed of two centrioles. This here, sorry, small thing. The centrosome is composed of two centrioles. Each centriole has 9 triplets of microtubules and a central axoneme, a doublet. And I don't know if you know this gentleman, Professor Albrecht Buehler. I was in touch with him some time ago. He was professor of cell biology at Northwestern University in Evanston, and by training a theoretical physicist like me, and he switched to biology, and he published a lot of work on cell recognition and detection of signals, and claimed that cells migrate to, at least the ones that he worked with, toward other cells by detecting electromagnetic signals sent by these cells in the neighborhood. And the two questions that arose were, number one, what is producing electromagnetic waves in a living cell? And his answer to that was, and others as well, mitochondria. So when cells are alive, energy is produced, ATP and GTP, primarily by mitochondria, and the byproduct of it is emission of near-infrared photons. As shown here, this is the peak of sensitivity, a wavelength of about 900 to 1,000 nanometers, which is near-infrared. As just a reminder, visible light is from 400 to 750 nanometers. This is longer, therefore lower energy, but and infrared. And then the second question he asked was, how do they detect it? And his answer was that this centrosome, which is shown here with these two centrioles, which are at right angles to each other, and they have these 9 triplets of microtubules, which are movable, a bit like Venetian blinds. They can shift, and he claimed by some argumentation they can detect light within 3 degrees in plane and out of plane. And then of course the question is what happens next? So light is energy, actually quite a lot of energy by biological standards. I don't know if you ever thought about this. The molecule of ATP carries something like 60 or so kilocalories per mole, which is about 0.6 kilocalories per mole is KT, thermal energy, which is given for free. But light in the visible range would be, if I remember correctly, about 200. But anyway, it's much more than ATP energy provides. So light is, to us, it's very subtle and almost doesn't do anything. I mean, you can feel in the skin in sunshine in the summer, it's warming, but you have trillions of photons hitting your skin per centimeter squared. But in living systems, we'll come back to it in a few minutes. Electromagnetic energy of photons are in small numbers, but each quantum of electromagnetic energy is substantial compared to biological energy. Okay, so this shows you, and we'll come back to it because there are some hypotheses that this light that comes out of either mitochondria as a byproduct or a reactive oxygen species as they recombine and they emit a photon that can be used by microtubules, in particular networks of tryptophans, which are amino acids that form a network on the surface of microtubules, can be captured and can be used for some form of maybe quantum processing. Okay, that's where the current thinking is, and a lot of papers, even recently, have been published on this topic. All right, so we go back to the neuron. We have the structure of the synaptic connections and the nucleus, and you have just schematically shown microtubules in the axon, but actually there are thousands of them, packing the axon, and the dendrites are so just okay. I'll come back to the dimensions. I was just tempted to say something about the distances and the sizes. I'll do that in a minute. But here you see Nancy Wolf from UCLA, she's now retired, published a lot of papers on the remodeling of this neuronal cytoskeleton as a result of learning and memory formation and things, not just long-term potentiation, which everybody knows in neurophysiology takes place when you learn and you create memories, but also things happen inside. And I just point out to these papers that Nancy Wolf published showing from laboratory experiments that microtubules are reorganized as a result of the processes involving memory. And we'll come back to memory. Calmodulin kinase, calcium calmodulin kinase 2 is an enzyme which is directly involved in memory formation. And also I want to mention that microtubules interact directly with ion channels, voltage-gated ion channels, and they can regulate them, block them through the presence of these protrusions called C-termini. Again, I'll show that in a minute. So that's a bit more realistic. As you can see here, my colleague Stuart Hameroff, has worked on the same topic for decades now, and involved a lot of people in the conferences he organizes every year. So that's a more realistic representation. And what you see here is the thick lines are microtubules. The thin lines between them are microtubule-associated proteins. And you can see here a very dense interconnected network architecture, which is quite amazing. And Stewart published I would say, visionary book in 1987 called Ultimate Computing, in which he predicted that this could be an architecture for some sort of computational process taking place, resembling parallel processing in computer chips. That was before we knew anything about microtubules, even before the structure of tubulin was revealed, by the way. So I read this book overnight. I couldn't sleep when I read the book, and that changed my life as a physicist, because I decided I'll study microtubules from that point on. And what this slide also indicates that almost all neurodegenerative diseases and neurological dysfunctions have something to do with the cytoskeleton in neurons. So Alzheimer's, as you probably know, is involved in MAP-tau, neurofibrillary tangle formation. So one of these microtubule-associated proteins, they're called MAPs. They detach and they form clumps or tangles, and that weakens the microtubules inside the neuron, and that causes lack of function, loss of function, and eventual death of neurons. So, but okay, that's another topic. It could be a separate seminar on what happens in various disorders, Alzheimer's, Parkinson's. There are, of course, other biomarkers as well, but microtubules or the cytoskeleton or motor proteins of the cytoskeleton always play a role. Okay, that's another view of the same thing, more or less. I look deeper in. And okay, these are the C termini here, which are important because I'll talk about this in a second. These little protrusions, 12 to 17 amino acids, long peptides, are hugely charged with electric charges, and therefore very sensitive to electromagnetic fields. And this might be a link to what Marco Cavaglia and Tommaso Firo talked about last time, where you have very strong electric fields propagating through the membrane in the form of action potentials. The electric field across the membrane is about 20 million volts per meter. This is incredibly strong. And electric fields propagated, so action potentials at about up to 10 meters per second. But importantly, that they reverse the polarity locally, and that can be felt inside this cell in the cytoplasm, and therefore these protrusions could be very sensitive to such changes. So I spent some time analyzing microtubules as resistor capacitor networks. This is maybe outside of this scope of this talk today, but I don't know. I have a huge number of papers, including experiments, which demonstrated that this is really what's going on. So you can, if you're a physicist, maybe if you're a biologist, this will be confusing. If you're a physicist, you can think about all of this as a massive network of resistors and capacitors. In fact, we've also found evidence for inductors being there. So RLC circuits, and then they have some resonant frequencies, and they can respond resonantly to certain frequencies of electromagnetic waves, and so on. So I'll just quickly mention these are the two papers that started us thinking along these lines. These are experimental. Well, this is theoretical, but one is experiment. The second is experimental paper with a lot of modeling papers that followed, showing two things: that actin filaments, in terms of electrical properties, actin filaments act as in terms of ionic wave propagation diodes, and microtubules, on the other hand, act as transistors. So they can amplify electric potential signals. And that's very interesting if you think about anything that can be amplified electromagnetically or electrically. And there's a lot of interesting stuff with some therapies, including, so-called tumor treating fields in the treatment of brain diseases, which kind of, on the surface of it, shouldn't work because they are very weak electric fields. On the order of about 5 volts per centimeter, something like that. But if there's an amplification mechanism, such as through microtubules, even weak fields can be propagated through the biological medium. So one message to the physicists here, as a physicist, we shouldn't think about live biological systems as inert media, and use something like dielectric constant of the inert medium composed of 70% water and some ions and stuff, protein. Because these are active, maybe even amplifying systems, may amplify signals. Okay. All right. So this is just a summary of, we've done many different experiments on microtubule conductivity. Even now I'm just finishing a paper on recent experiments to quantify the values of resistance and capacitance and inductance. If anybody here is interested, I can give you the numbers that we obtained from experiments and theoretical modeling. But again, think about a microtubule as a by biological standards, a giant RLC network. So resistor, inductor, and capacitor. Why? So capacitor, why is this a capacitor? Because you have a lot of negative charges on the surface of a microtubule, which attract counter ions. So you form a cylindrical capacitor. It's a double layer. So that covers the C. Why is this a resistor? Well, because, and this is interesting, it's not only a resistor, but it's a variable resistor. The value of the resistance changes depending on the medium. It beats semiconductors because this is a very dynamic electrical system. Resistance because the ions can flow along the microtubule, and this is kind of a conducting part. So yeah, that's they aggregate. Some of them condense and form the capacitive part, and some of them are free to flow. But this is a lower dimensionality. Now they have a one direction to follow, and conductivity of microtubules at low ionic concentration exceeds that of the medium, even by a factor of 1,000. So they become wires. When you increase conductivity, when you increase the concentration of ions, this changes. They become less conductive than the medium at some point. And we published this in Frontiers recently. So it's variable, it's tunable. You can think about, this is super smart. The cell can either use them or not, switch off and on, depending on, let's say, concentration of potassium or sodium, and so on. And finally, why is L? Where the heck would L be? Well, because this has a natural, microtubules have a natural helical structure. So you can think about some ionic flows wrapping around like a solenoid. Now you have a solenoid. So if you have a straightforward flow, you have a conductor or a resistor. Yeah, either way. Some of the charges are stuck. They are attracted to the negative. You can't pull them out of it. but then you have a layer that would be forming a solenoid around it. And once you have a solenoid, you can think about all kinds of interesting things, like interactions with the magnetic field, right? Because now you have a magnetic field effect and electromagnetic and so on. So this is a huge area which is, I would say, ripe for investigations from the biological perspective, but also technology. Nanobiotechnology could be implemented here. And you know, these if you think about these circuits in neurons, they are very regular and stabilized by maps, so almost immutable. But in other cells, microtubules are assembled, disassembled. They form different architectures. And if you ever studied Kirchhoff's laws for circuits, I don't know if I'm using a foreign language to you guys, but anyway, then you know that the effective resistance of a network changes dramatically from series to parallel. In series, you add up resistances, in parallel, you divide. And if you have something in between, you can regulate the effective resistance of the network. So from the point of view of architecture forming, microtubules are just amazing structures. This is a microtubule, by the way. Computer reconstruction, and you have a cross-section here. And these little things are C-termini. So this is where 40% of the electrostatic charge is. They are very flexible. In fact, they have not been resolved crystallographically due to their flexibility. And being flexible, they are just like antennae. So they will be responding to electromagnetic perturbations very rapidly. Electric charges, each one has about well, let's say, so 40%, a dimer has 50 charges, 2 C-termini, so 25 charges per monomer. So about 10 charges, 10 elementary charges, negative. Okay, this is all negatively charged. And one more twist, if you get bored with electricity, tell me. But one more twist is that the amount of charge on the microtubule can be controlled by pH. So that's another weapon in the armory of a cell. If you make it high pH, you increase the negative charge. If you go down with pH, you reduce the charge by protonation. So histidines get protonated. And actually, we found in our experiments that at pH 5, microtubules are neutral. They don't have they have no net charge. Below, it's even positive. So interesting stuff, to me at least. This is a surface of a microtubule. You wrap it around, make him. So that's unfolded. Let's say it would look like a carpet, but then you fold it and make this this cylindrical structure, but it has a helical property around. So it's a helix wrapped on a cylinder, but complete. They have and hollow cylinder. So in the middle you have a lumen, which nobody knows really why this is so. Some people speculate this is a channel for safe transport of some material, shielded from fluctuations outside. And then you have these C-termini. This is an alpha and beta monomer. That's a building block of a microtubule. Okay, and it's all dynamic in cells other than neurons that stabilize them. You have polymerization, depolymerization phases called dynamic instability. It's all driven, by the way, by GTP, so energy flow. It stops if energy stops. Microtubules become static. You need to constantly supply GTP. So it's a non-equilibrium process. And that's the only such thing so dynamic instability is the only such thing known to me in biology, as far as I can tell. Okay, and that's structural change on hydrolysis. So from a straight protofilament, you go to so that's 13 protofilaments make typically one microtubule, and it has to have GTP attached to beta tubulin to maintain this straight conformation. When GTP hydrolyzes spontaneously and stochastically to GDP, it curves. And that is the reason for instability. The structure then collapses like skyscraper dynamited with sticks of dynamite. These are the sticks of dynamite, GTP, that go to GDP. It's the biological energy explosion. It's not a lot. It's just over KT, but enough to. All right, so I'll skip this. Okay, so so why would this have anything to do with memory? Well, we'll come to it in a second. So in addition to all this electrical stuff, there is also a great potential for the storage of bits of memory. And this is just a history of, on one slide, of people thinking about how much memory the human brain can have. And John von Neumann, the godfather of computer science and a great physicist, quantum physicist, he actually was the closest to the truth, in my opinion. He predicted the human brain can contain 3 times 10 to the 20th bits of information, which is quite enormous. And I kind of agree. You'll see why. And some people offended, in my opinion, the brain by giving these small numbers, including Thomas Landauer in 1986. How do people remember? He said that we retain about 2 bits of information per second. So over the lifetime is about a gigabit. Now a memory stick they can buy for, I don't know, $15 or something has terabytes, right? Terabytes of memory on a memory stick. And our brain that is so powerful would have 1 gigabit. I mean, come on. Okay, this is just to remind everybody here what, I don't know if Mike Levin is here, but you know his work and his fantastic work on the what is it called? These worms. I forget the name now. Decapitation of the worms.
Planaria.
Planaria. Yes, exactly. So we are still searching for memory storage, but I propose that memory actually can also, maybe in addition to something else, long-term potentiation, maybe stored in microtubules. And this is what we published some time ago. We know that, as I mentioned, there is cytoskeletal remodeling and learning and memory. So very quickly, because time is running out, there is a potential for massive amounts of memory stored in microtubules. So first of all, C-termini I showed you, they can be in at least 2 states. Once we showed it computationally, one state binding to the surface, one sticking out to the outside, and maybe intermediate step, let's say 4 per dimer, 2 C-termini, 2 states, 4 per dimer. Then there could be electron hopping, as we know from some simulations and even some experiments. And so this is, and then conformational states, GTP regulated, GTP, GDP, 2 conformations. So it's 4 times 4 is 16, times 2 is 32 per dimer. Each microtubule has about 10 micrometer microtubule have about 16,000 dimers. So if you do the addition and calculate the number of microtubules in a neuron, you end up with 1 gigabyte per neuron, not per brain, per neuron. If you have 10 to the 11th neurons, that gives you 10 to the 20th, on the order of 10 to the 20th. Remember, for John von Neumann, he said 3 times 10 to the 20th. That will be just maximizing the use of your microtubules in neurons. Maybe some other things, but that would explain. So, and then the question is how fast you can change these states. Depending on what you do, it can be fast or slow. Protein conformational changes occur in nanoseconds to microseconds. So if conformations occur like that fast, you could have a very rapidly functioning supercomputer in your neuron. So each neuron would be a supercomputer. But that's too fast. And I will not get into the great details, but there is something called a different Landauer, not Thomas Landauer, Rolf Landauer, came up with this Landauer in a theorem, which says that each bit of information costs energy. A minimum in physical terms is kT times logarithm of 2. But biological energy is more costly. It's ATP or GTP, more than kT. So if you do this calculation, there is a limitation on the number of bits per second that the brain can consume with the, you remember I told you in the beginning that's about 5 watts per information processing, right? The brain uses 25 for everything, let's say 5 for information, cognitive processes. So if you take the 5 watts as your energy budget for cognition, then you obtain a maximum number of bits that you can process without your brain exploding due to overheating. Okay, and I did this calculation. It depends on the speed of changes in the states. Maybe that's inside baseball for most, but anyway, if anybody's interested, I can show you the calculation. So back to another form of memory storage, and this is actually backed up by some experiments, and this is now 2008, Dr. Tien, I think that was NIH, they did experiments with mice, and they used calmodulin kinase 2 inhibitors and demonstrated that when the mice are given these inhibitors for the enzyme, they forget how to solve the maze. And it's reversible, right? So that's so what's CAMK2? Well, CAMK2 is an enzyme that phosphorylates tubulin, and in fact phosphorylates in the C-terminal region, which I showed you, serines in the C-termini. Okay, and this is how it works. It's activated by calcium and forms these hexagonal motor. It's a motor protein, actually, of hexagonal motor proteins. It's a hollow enzyme. And so we looked at how this can work, and it shows you the surface of microtubule. The turquoise dots are C-termini. So this is all the places where phosphorylations may occur in microtubules. And the red and blue diagrams are electrostatic charge. So red is negative, blue is positive charge, mainly negative, okay? So now this is that enzyme. Travis Craddock, my student, now professor, that ages me a huge amount, but anyway, reconstructed computationally this CAMK2 enzyme. It looks like this, different views, okay? So here's just a colorful representation. Sideways, it looks like a transformer with legs and walks on microtubules. And from the top, it looks like a snowflake, right? It looks like a snowflake. And here you have the charge distribution painted on this enzyme, and beautiful, right? I mean, it's beautifully symmetric. And then we demonstrated that it actually fits beautifully also to the surface of a microtubule. So that's to scale the size of the enzyme, and this is this patch of the microtubule surface. And this shows you how it fits to it. So geometrically, it makes a match. So it lands on the microtubule, like shown here. This is a cross-sectional cut, and walks on it. And as it walks, it phosphorylates C-termini. This shows you also that there is electrostatic attraction. So it is matching positive charges on the enzyme matched to the negative charges on the tubulins in the microtubule. And then you have this little poodle, little dog walking. I have a dog right here, a spaniel, and walking and phosphorylating and leaving information. Phosphorylation, as you know, is one of the ways of leaving information. So we did the calculation, and okay, that's another view of this. We calculated that each calmodulin kinase can encode up to 52,000 bits of information on a microtubule. And then you do the math, calculate how many microtubules, how many enzymes, and again, you end up with another gigabyte per neuron. This is different from the other estimate, by the way. These are different processes from the ones I told you about. So we can double it. Now we have 2 times 10 to the 20th. Von Neumann said 3 times. So we are still looking for another 10 to the 20th of bits. I'm joking, but we're in the same ballpark. So that shows you kind of a pattern that could be left on the surface of microtubule. Red would be phosphorylated states. And then you count the number of states, you get that. And somebody asked, what would this be good for? What sort of information? Well, the information could be, for example, to make attachments to maps or detachments. And that would, in other words, it's like traffic signals, yellow, red, green, and you can change this and you can rebuild the highways. As I told you, microtubule networks are used for intracellular transport. The simplest way would be just to regulate transport through it, open some lanes and close some other lanes and allow for transport between microtubules and things of this nature, or maybe electrical signals. Either way, you can create logical gates out of this. And that shows you these little kinesins walking and then finding an obstacle, and yeah, they have to change the lane because it's a red light. You can't go there. Okay, that's the paper we published now 14 years ago with Stuart Hameroff and Travis Craddock. He was at that time a PhD student. Okay, so maybe I don't know, I have more, but this shows you how these microtubule associated proteins maps can rearrange themselves and form different connections and architectures. And as I told you, the architectures change the IV characteristics, electrical properties, and so on. So yeah, so let me just skip this, and I'll try to finish in a minute with something that probably goes back to the beginning. Yeah, so quantum. Is there any potential for quantum interactions? And the answer is yes. So far, everything was classical, by the way. It's either mechanical, electrical, electromagnetic, but not necessarily quantum. And this is, what Penrose and Hameroff proposed. These two transitions from GTP to GTP state of tubulin involve conformational change, but they may also involve a change of the electronic structure and the quantum state. So that's the postulate. The thing that I completely disagree with is that this is spontaneous and that this is gravity induced by some way. I think gravity plays no role, in my opinion. It's too weak a force. So we found that that tryptophans in tubulin form complexes very much similar to FMO complexes in photosynthetic systems. Okay, here you have the network of tryptophans, and tryptophans can be photoexcited. This is the only, well, there are two other, but the easiest amino acid that can be photoexcited. So you have 8 tryptophans in a tubulin dimer, and you can now imagine a network spanning the entire microtubule, and then you have other microtubules with the same network, and they can start creating a quantum state. Okay, that's the hypothesis. We published this prediction, feasibility of coherent energy transfer microtubules, in Journal of Royal Society interface some years ago, and 10 years later, roughly 8 years later, we did experiments showing that this is indeed the case. So that was the illustration of the pathways. Let me just, that answered the question where the energy could be coming from. I told you about mitochondria and reactive oxygen species. Let's skip the anesthesia because there's no time, and also this. Okay, so these are the experiments, biophotons. So biophotons are electromagnetic waves emitted by all living systems when they are alive. Okay. And in these experiments, we use the intensity corresponding to biophotons. This is the paper with Greg Scholes, by the way, who is one of the godfathers of quantum biology. We did experiments at Princeton in his chemistry lab with my student Arat. And that showed that we are getting somewhere with this. These experiments show the absorption and then re-emission of photons by microtubules on the time scale of tens of nanoseconds. Now, half-life is about 5 nanoseconds maximum, but they can persist up to tens of nanoseconds. It's short, but it was in disorganized microtubules, by the way. So it's not like a neuronal structure architecture, which may increase the lifetime. So, and also we compare to tubulin. So if it's microtubules disassembled, these states are shorter lived, about 2 nanoseconds instead of 5 or more. And then with another molecule called AMCA, we were able to quench some of this and find out at what point the quenching stops. And it turns out that this corresponds to the size of the what we call coherent domain. something like this, right? So when you shine light on it, this creates a quantum state which couples all of these tryptophans. They are in a single quantum state. They're correlated quantum mechanically. And that basically is the size of this coherent domain, about 8 nanometers, 6 to 8 nanometers. So the entire dimer can be in a single quantum state. However, as I said, only for about 5 nanoseconds, maybe 20. Physiologically, we need something like milliseconds. So we still have a big distance to go. So I will stop at this point. Just maybe one more comment. And this is with anesthesia, which is, a mystery still after 150 years or so of being used clinically. And the current understanding is that the Mayer-Overton graph explains anesthesia, namely, the more lipophilic the compound, the more potent an anesthetic. But we found actually that anesthetics bind to tubulin, and here, so the red dot, so blue are tryptophans, the ones that get quantum excited. Anesthetics are red. These are the points where anesthetic molecules bind to tubulin. And we predicted computationally that this would actually quench the quantum state. In other words, the quantum states of tubulin would be either shorter lived or canceled completely. And that's the link to consciousness. The closest you can get in the lab, I think, without working on live neurons or brains. So, okay, I'll stop here. And if you have any questions, I'll be happy to answer.
Jack, that was fantastic. Thank you so much. I could listen to your presentation all day. I was just like, I have 1,000 questions on every slide. Yeah, so we'll go into the discussion part of the meeting now. Just a quick note, if you're not muted, I may mute you at times. If someone else is talking, if you have a question, raise your hand and it'll show up in my feed in order that you raise your hand, and I'll just call on you. I usually start off with a couple of questions, and then we just go into free discussion. Let's see, Jack, if you could stop sharing your screen.
Oh, yes, of
that
course.
would be
I was
good.
wondering.
Yeah,
I was wondering about it. Great.
awesome. Yeah, so I guess just to sort of kick things off with questions, I just wanted to kind of repeat what I heard in general and see if I'm hearing this right. So, microtubules, they're basically universal to eukaryotic cells. They're not so much found in prokaryotes, but basically across eukaryotes. And then they've got they're electrically alive. They're capacitors, transistors. They have this enormous potential information processing capability. And then, you know, that's so cool that it seems to correlate to von Neumann's predictions. That's
Yes.
cool. And yeah. But anyway, but then I heard you say basically my understanding of what you were saying about CAMK2 and tryptophan and pH was that all three of those are different modulators and they're reversible potentially.
Yes.
And okay. And so that information isn't necessarily erased when it's modulated by those.
Right. So that raises the question of, yeah, of course you can erase information in various ways, by overheating, thermal ways, fluctuate, or
Right.
enzymatically. You have kinases. CAMK2 is a kinase, or phosphatases, so it could probably remove this phosphate group enzymatically. And some people ask me, well, if that's the case, and microtubules, by the way, during your brain's existence, they are remodeled themselves. There's a turnover of tubulin. I don't know, after 6 months or so, a new tubulin is added and some is taken out. So how would the brain retain information? Maybe it loses some of it as a result of this during aging and remodeling of, and of course, yeah. So that's a valid question. I don't have a good answer. I think maybe there can, just like DNA repair enzymes, maybe there could be some enzymes that recode. If you remove a tubulin, then it comes back and recodes what's been taken out. I don't know. I'm just fantasizing at the moment.
Yeah. So I have a follow-up to that, which is, obviously microtubules in the cell are organized in a really specific way, in a particular structure that, like, if your interpretation is correct, it encodes some sort of cellular memory of how to do things. And my question is, if microtubules are these general information processing structures, can you think of experimental ways to talk to them, like to communicate, to read what they're saying,
I
to
see.
communicate back to them? I mean, is there a, outside of the context of cell biology, if you use microtubules in a way that you know preserves or it somehow does justice to their structure without just blending them randomly, um, are there ways to sort of test that hypothesis that they're storing information? I'm just throwing that out there as a wild idea.
Right. So one more comment before I answer a question, that I would consider all of, the things I said, maybe sounded a bit confusing, but I would consider many of these things as independent channels of communication that may be used for different purposes. Some may be used for immediate action, some may be for storage of information long term, and some for intermediate. So you may have several channels. It's like a cable, coaxial cable with many. You can watch on TV, I don't know, 500 channels, switch channels, because they're on different frequencies. The same with microtubules. It could be ionic, electronic, protonic information, or electromagnetic. All of this is in principle possible. Okay. And then the reorganization of microtubules may lead to some larger scale, so hierarchical, passage of information or different level of information. And for me, this, by the way, is one of the key questions in biology: hierarchies. How different hierarchies communicate with each other. And here we have the lowest hierarchy is the tubulin dimer. It's its own, dynamics. And the next one is a microtubule. The next one is bundles of microtubules inside the neuron, and then neurons talking to other neurons in the vicinity or across the brain. So that's the, to me, $1 million question. Maybe not the heart problem according to Chalmers, but for me it's still a heart problem. Now the question is, yeah, how do so read the write, right? This is we know how to write information, and you want to see how you can read this information, right? That's, if I understand your question. So writing, as I said, writing can be just by passing signals, electrical, electromagnetic, or chemical signals, and or even mechanical. Oh, by the way, I forgot to mention, back in the 1990s, Donald Ingber at Harvard showed that microtubules pass mechanical signals from outside the cell all the way to the nucleus. So I forgot to mention mechanics. That's another thing.
Right. Well, and
So
it's sort of a circulatory system as well, right? I mean, that's a component of what it's doing. I mean, it's not just electrical.
yes. Yeah, it's mechanical, electrical, chemical, electronic, electromagnetic, maybe even magnetic, with the solenoid aspect. No, and the possibility of switching between different modes is kind of intriguing, and that's not what you have in silicon based electronics, right? You build a circuit and it stays this way. This is a cell creates its own circuitry, which is evolvable, and changes depending on demand. If it wants to divide, it'll reorganize microtubules. It will move mitochondria away from microtubules. That's one thing which is kind of interesting, and so on. Now, how do you read this? Okay, so that's the hard question. I think the simplest possibility for me would be doing basically what we did actually. I didn't show you the experimental results from some of the more recent work, which is IV characteristic, So you inject a current and you measure the voltage and you calculate the resistance of the system. And that's analog computing. So you can create these resistor networks, and the signals, electrical signals, it could be either, just like you send, signals through the wire, electrical signals through the wire. In the olden days when you had, the lamp-based technology,
Right.
So you can signal amplification of something, and maybe making a protrusion. Maybe the signal is for the cell to crawl or to shrink. All kinds of things. And this is what Albrecht Buehler was talking about. I don't know what happened to him. If anybody knows, I'd like to find out.
Well, that's a great segue to Mary. So the questions in order: Mary, Richard, Henry, Cy, and then Natalie. Mary actually studies with Albrecht Buehler. So go right ahead, Mary. You're still muted. Mary, can you hear me?
Sorry, my computer froze. I'm in a very crazy environment. You can see the construction tracks behind me. Albrecht Buehler was a really interesting guy. I studied with him at Woods Hole, and then later I started in the late 90s. I was trying to assemble the cytoplasm intelligence
I have a bad connection.
that he talked about, and I was studying just the chat, Jack. Maybe I should just send them to you, but here's one. Is the capacitive behavior of microtubules dependent on organized water on their surfaces, easy water? And if so, does disrupting it alter the transmission properties of the microtubule network in ways that would precede, for example, synaptic failure?
Yeah, the answer is yes, for sure.
Okay, great.
Because the capacitive ability of microtubules is dependent on the thickness of the layer between the charges. And
Yes.
if you have easy water, then you'll change the thickness. I haven't thought about this, but this is a brilliant question.
So I have a bunch of things to send you that I've been working on for a while.
Please, because that stimulates my brain cells.
Okay, last year I was talking to Marco about the possibility that the membranes were lamellar and they were creating a holographic plate. I should catch you up to that whole. In my biology framework, I mean that organism scale coherence is maintained by the integration of piezoelectric signals from structural proteins through the exclusion of the zone water that coats every biological surface. So, am I still frozen?
Occasionally.
We're having little glitches in your
Off and on.
in your audio, but your questions are there in the chat, and so we can all see
well,
them.
the thing is, in our calculations of the properties like RLC for microtubules, they all depend on the length. They all depend on the ionic concentration, and that affects something called the Debye length. The Debye length is the distance over which electrostatic interactions are not disturbed by thermal fluctuations. So the easy water will change it, and of course, ionic concentrations affect. There is a formula, and I will refer you to a paper we actually presented in great length.
Great.
This was triggered by experiments. I couldn't believe it that at low ionic concentrations, we found microtubules to be highly conductive, like, as I mentioned, 1,000 times greater than the medium. And then we went to physiological concentrations, and suddenly their conductivity dropped like a stone. And I said, what's going on? And so that led to this the Debye length calculation, and it's Poisson-Boltzmann equation. You have to solve the Poisson-Boltzmann equation under different
So,
conditions.
so that makes me think that the easy water is like a proto, the dielectric phospholipid membrane, in that it slows down the protoneuron in the way, sort of a parallel there. I'll let other people ask questions.
I think you're right.
Okay.
Great. Richard?
Thank you. Jack, that was really fascinating. I had some inkling that microtubules were a little bit more than scaffolding poles, but I had no idea of the depth of elaboration you were going to tell us about. That's really fantastic. Right back at the beginning, you mentioned microtubules being an experiment done with microtubules and the cell changing its direction towards a pulsing light. And you talked about the frequency of the light, but you also mentioned that the light source was pulsing. Can you tell us more about the frequency of the pulse, or why that might be significant, or your thoughts about that?
Right. Well, okay. So that wasn't my experiment. That was actually
Mm.
Günther Albrecht Buehler's experiment. Maybe Mary can comment. But in general, the pulsation elicits biological effects much easier than constant fields. It applies not only to light, electromagnetic waves, but also to electric fields. We've done experiments, and that's another topic, and many papers on experiments. We actually experiment right now with the frequency of pulsation. We use near infrared device, which is used for photobiomodulation, therapeutic effects on the brain, and there seems to be a sweet spot. I think if I remember, because we tried low frequencies like 5 hertz, 10 hertz, 20, 40 hertz, and 100. And I think at about 10 hertz, we have the best response. And that has to do, by the way, we also noticed that it affects the functioning of mitochondria. So there's a lot of stuff going on, not just microtubules, but energy supply driven by pulsation of electromagnetic waves. I
Haven't
think,
tried Mozart yet. So
and I'll
you
be happy to
mentioned,
send some papers we wrote, on these things. I didn't really go in depth because
yeah,
you
sure.
guys be sitting here for 20 hours and I'll be talking like an idiot.
We would be glued to it. Thank you. So you mentioned the RLC circuit's potential to act like an
Yes,
antenna,
exactly.
uh, and it's not just a simple antenna that would potentially just respond or carry signals on a single frequency, but that it would respond to a combination of frequencies, right?
Absolutely. And in fact, it's another layer of amazing complexity that the cell has at its disposal. C-termini are the primary area for post-translational modifications in cells. And there's a whole repertoire of post-translational modifications which will change the mass and maybe charge, depending acetylation, methylation, all of these things. There's a huge review paper by my colleague Richard Ludueña about post-translational modifications of tubulin. So again, the cell may tune to different
So it's
frequencies
right
by
in there in
changing
in
the
the
antenna. It's
genetics
like
as
your
well.
dial on the radio, old
Yeah,
fashioned.
exactly. So, so you know that the RLC circuit created by the microtubules, through its three-dimensional structure and the connections between them, would tune it into a particular frequency to receive a sensitivity to a particular message, not just tuning it into a particular frequency. And uh, the microtubules dynamically reorganize and restructure as a result of the signal they're receiving?
Well, okay, that's again maybe more intricate. Could be, but okay, we did experiments on this too, and I didn't show you. But we showed actually that with specific frequencies, it's in the low terahertz. This is now we're talking 10 to the 12th. If you go through the airport scanner, this is the frequency that you encounter, your body encounters, you don't see it. After 15 minutes, so you're safe, 5 seconds is not. After 15 minutes, we disassembled microtubules completely. And I have movies showing you how they disassemble in 15
Yikes.
Don't stick your brain for too long.
So,
My God.
so the kind of feedback that I was interested in, so I'll see if I can put that together a little bit to come to my point. I'm imagining a communication that is picked up by a cell with a particular microtubule structure that reformulates the structure of the cell to become a transmitter of the same song. So that so that the way in which information could be stored and recalled is through the structural reconfiguration of the microtubules to produce the same song that they received, so that they become like a longer term memory of the electromagnetic signal that was received.
Beautifully said. Yes, I couldn't have said it any better. That's
Very,
exciting.
very exciting. Thank you so much.
Great. Henry?
Oh, hi.
Hi,
Enjoy your talk.
thank you.
So I have 2 or 3 questions. So the first question is, you actually did the very deep story of the link with this information capability with the microtubule. But is the same story can be true if we do the same thing for the mitochondria, for the membrane potential?
Maybe not. Maybe not the same story, maybe some of it, but I'll tell you why not the membrane, because the membrane is, first of all, microtubules are the most regular, most symmetrical, and most rigid structures in the cell. Actin has similar rigidity, but it doesn't have the same intricate geometry and the size, right? So
Actually, I agree with you. Analysis. Only thing is, pre your work, we read lots of the mitochondrial link and the membrane link. So they actually also can write books as such.
it's
So
linked, but I don't think mitochondria per se. Well, then I haven't studied enough. I only know that they definitely associate with microtubules in different stages of the cell cycle, and they provide ROS and all this stuff and ATP.
Sure.
But in terms of structural information and coding, I'm not so sure how this would be. Okay, I simply don't know.
Okay, so I agree with you because I think the different molecular or biological physiological phenomena we need to prioritize in terms of contact. So in such a dynamic regulation that contact, I agree with you. So so my question is for a while we're thinking the information as a package, we're always thinking at the system level. For example, the system level of information, including how many microtubulin, assembling time, interaction with mitochondria. So those, as you just mentioned, is controlled by another hierarchy of the order. So we actually thinking the system information. Basically, for example, if you use a cell as an example, we always thinking nuclear. But what you have said is fascinating, is the parts of the information process actually is highly dynamic and replaceable. So that is something fascinating because system information you cannot replaceable. This is species specific. So they're just generation by generation the same. But with such a potential, the parts information can simply can amplification or can even create new information. But such information do not need to be inherited. They just solve the current problem by the environmental dynamics. Is that correct?
Yes, absolutely. This is indeed, I would say, epigenetic information, right? I mean, there is a genetic aspect too, by the way, which I didn't go into, because there are about 20 genes encoding for different tubulins, and there are some, of course, mutations, and there's isoforms of tubulin, and every cell expresses different isoforms differently. It's another repertoire of possibilities for the cell. It's, alpha 1, 2, 3, 4, 5, 6, 7, 8, beta 1, 2, A, 2B, blah, 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 isoforms. And they can construct, it's like you can build a mosaic from different tiles of different color. So each tubulin dimer is a different color of the mosaic that put in the mosaic. And then on top of that, you have these post-translational modifications. So that's epigenetic. For example, they can be associated with different diseases. Different modifications occur in cancer, as is well known, and so on. So, I mean, this is a combinatorial explosion of possibilities. The cell, yeah, can use. And then the system, that's another thing, right? That's on next layer of information for the system.
So just to follow what you say, therefore, actually, there's no point for us to really try to figure out how the epigenetic, the information can be inherited. It doesn't matter in such a sense. Is that correct? This thinking that way.
I don't know if I can say like a blanket statement like this, but definitely much of it, yes, is not. It is just a response to environment. And by the way, I'll give you one example from biology. We did experiments, and I think we published that some time ago, because we targeted a specific form of tubulin, isoform of tubulin for inhibition, right, in cancer. And we designed drugs that were doing this. And then we did experiments with cell lines, and then we measured expression. And different cell lines either stopped producing this particular isoform which was targeted, and other ones overproduced. So they decided to make up for the difference. I mean, that was amazing, fascinating strategy, right? Different cells will have different strategies to deal with stress. In this case, it was a chemical stress by using this toxin.
Okay, thanks. Yeah.
Well,
Amazing. Sai?
Hi. Yeah, I have a question that's actually following up on something you just said about genetics. How constant has mitotubular structure and function been over evolutionary time? And, things like the amino acid sequence of
yes,
tubulins and things like
perfect question. I mean, amazing. 2 billion years. It's almost, your tubulin is almost identical to that of a paramecium.
Okay. That is incredibly important.
Yeah. And the differences we studied, actually I published papers, we've done phylogenetic analysis of all of this, but the differences in tubulins between species, and across evolution occur in C termini. The C termini are your address. That's the rest is structurally stable and sequences are conserved.
Wow.
But the 12 to 17 amino acids in the C terminus give you the origin of your tube. A friend of mine actually commercialized this information. Can you believe? He developed a technology, a biomarker to find if, let's say, if the meat that was analyzed came from that particular, farm, or in fact, he's Italian, so he was able to detect which wine came from where. In fact, even analyzed some wine from Roman times. It's so precise, and you can do it. It's 20 amino acids, right? So it's 20 to the power 17. It's an astronomical number of possibilities of encoding in just the C terminus.
Thank you.
Well,
It's incredible. Hi, Natalie. Welcome.
Hello, thank you very much for your presentation, Jack.
Sure.
I don't know how familiar you are with Lynn Margulis and her theory of the serial and the symbiotic theory.
Tell me. No, not much. No, I heard the name, but I haven't really.
So
studied.
Lynn Margulis assumed that she tried to put a chronology on how mitochondria evolved from bacteria and how plastids evolved from cyanobacteria. And she also assumed that there was a primary endosymbiosis between spirochetes and archaeoplasma. And she assumed that that was something that was responsible eventually for the origin of the eukaryotic nucleus and for the orchestration of genes in chromosomes, of mitosis, etc. And so one of the things that she did there was compare the centriole kinetosomes that you were talking about with the tails of flagella and with microtubules in general. And she assumed that there might have been a bacterial origin coming from spirochetes for microtubules. Is that something that is being researched? Is that something that is being
Possibly.
taken? Yeah.
I mean, yes, I don't know much about this, but I know that bacteria produce not tubulin, but FTSZ. It's a bacterial analog, which is the sequence similarity is not that great, about 40%, but structurally is quite similar. So there's higher amount of structural similarity, but they are not forming microtubules. They form filaments like actin. So something like that. So tubulin is the only one that can produce microtubules. I don't know about the origin of these other species, but it's kind of interesting to compare and see if that's a a good explanation for how things evolved in early planet life.
Thank you.
You're welcome.
Amazing. Awesome. Mary?
Hi. This is a shorter question. Could microtubules be helping to formalize or transmit memory and an electromagnetic signature and then transferring it to a slower medium for long-term storage and future readout? This isn't my whole theory. I'll send it to you.
Okay. I don't know. I have to think about it. I will take a fifth on this one, but it's a good question.
Thank you.
Nice.
By the way, just something came to my mind. Possibly because they also form larger structures, right? So maybe
Yeah.
higher level organization could be informed by these things.
Well, actually, that relates to I think that the electromagnetic coherence of the organism is mediated by these structures interacting with, like, in particular, tubulin and probably collagen, and mediation relationships with structured water. And this relates back to Mei Wan Ho and those Italians who worked on easy water.
I see. Del Giudice,
Yeah,
I
Del
know
Giudice,
them.
yeah.
He passed away, unfortunately, some years
Yeah.
ago.
Interesting guy.
And Giuseppe Vitiello, he's still alive, as far as I know.
Yeah.
Yeah. Awesome.
Could I
Yeah.
ask one? Oh, sorry.
Oh, go ahead. Go ahead, Henry. Yeah, I was going to take it off in a different direction, so go for it.
Okay. So seems like for example, microtubulin. People ask the evolutionary question, but if we think in the evolution is a mechanism which just capture the incident or miracle and change it to a biological thing. Therefore, every cell dividing have microtubulin, just like a mitochondrial story, right? So therefore, it's no longer evolutionary story. The evolution story is how the microtubulin further were combined with the brain cell, neural system, and merge to new system, then be captured again. So if we think in that way, actually I think is fascinating is because of quite turnover rate of many of the parts.
Yeah.
Information process actually is just a biological issue. For example, you have so many microtubulins that quantitatively they can regulate, and also you mentioned the topologically combination, everything that actually is a biological issue, right? So maybe it's no longer be controlled by the nuclear genome even, because you are the system, you already have this. Is like you have the organization, you already have janitor. You do not need how to regulate the janitor's work, right? So in that case, we're actually thinking complexity. Therefore, the microtubulin in the brain cell could be very different from the bacteria similar system, even though they have the same amino acid. So in that case, how should we actually
Yeah.
use evolution ways to understand this complexity? A
lot of questions, good questions. So number one, I think that because microtubules have so many possible functions and properties, I think over the course of evolution, these functions may have been discovered by different cells. Oh, I can use it for this. I can use it to create centrosomes. I can transmit. Now I can put them together and make a neuronal scaffold. This kind of thing. So, of course, the brain microtubules. It's higher order organisms, right? And the second thing is these isoforms. Just to put, beta-3 tubulin, which is one of those that I studied, is almost exclusively expressed in two organs, the brain and the testes, for some reason. So again, different genes become expressed or activated for different purposes, and maybe different types of tubulins are used for just like you order different types of material to build your house. You use wood, a brick, or something else, concrete. And whatever you need, you use. That's kind of my interpretation of this thing. I'm not an evolutionary biologist, so I don't really know that much, but I know how much can be utilized by all the various forms of tubulin and types of assembly of tubulin. And by the way, there is also, under various conditions, and this is more technological than biological, you can make, out of microtubules, you can make sheets, you can make spirals, you can make macrotubes. So this is an incredibly universal material for all kinds of constructions.
Yeah, thanks.
Awesome. That's so interesting. So I was going to take the discussion in a potentially complicated direction. It's already pretty deep. So in this group, we basically discuss, debate, hypothesize different ideas about cellular cognition. And as we do that, we kind of have two elephants in the room at all times, which is that we're looking for these universal features of cells and cognition that don't necessarily require consciousness, although they might explain consciousness in some cells. And some people think that all cells are conscious in some way. And those ideas are welcome here too. But it's just consciousness is kind of a subject that we a little bit try to do without. And the other elephant is quantum effects, because because everybody sort of has a hard time thinking in quantum terms, and it's kind of a dangerous territory to begin, like what's already an incredibly interdisciplinary and synthetic project to even conceive of cognition in the first place, to then put it in quantum terms. But I noticed that you and Marco and Stuart Hameroff have not been afraid of those territories. And I guess I would just ask you to, comment a little bit on how you feel that these ideas relate to those.
Okay. So first of all, I don't think it's all or nothing. So I think I don't see microtubules as necessarily quantum devices, but we definitely see the potential for some quantum processes being taking place there. So it's largely classical, it's biochemistry and cell biology, but then there's a layer of potential quantum communication. So it's another channel. One channel of communication could be quantum, using these tryptophans and excited states that may expand across a microtubule, perhaps, or even between, if we create architectures and conditions as well. So that's that. And it's a highly interactive thing. So I didn't really have much time to emphasize the connections. I mentioned the connections to ion channels, to membrane, to action potentials, and all of these things play a role in the dynamics of microtubules and all the information storage and processing. I just wanted to say that the potential for information storage in microtubules is unparalleled. I don't see any other biological system that can do it. And membranes membranes play a different role, in my opinion. They are really the communication relays. Of course, with action potentials, electric fields, and then neuron to neuron transmissions. And microtubules may affect this, and may do two different things. Number one, could store information. For example, each passing action potential may leave a trace on a set of microtubules. And then I remember, okay, that was a stimulus. Somebody pinched me, somebody told me something, okay. Then I have encoded this in the microtubule due to the signals that were propagated. And then there's the reverse. Microtubules, by interacting with ion channels, may affect the production of electromagnetic waves through membranes that Marco and Tommaso talked about. So there is a two-way street. I don't think it's just so segmented. And one thing which I wanted to mention in this connection is the abnormalities in microtubules. For example, we talked about Alzheimer's, maybe even schizophrenia, some other things, may be reflected in these distorted images that Marco talked about. The holographic images may become distorted as a result of these interactions. So there could be either genetic mutation that causes a distortion in the image, and then you have a different view of reality. You may have schizophrenia. That may be also not genetic, maybe sporadic, and so on. But anyway, I see a potential for making a connection between these things. I wouldn't take them separately, because, as we argued, 2 billions of evolution on this planet, and conserve something so versatile. Why would you throw it away? Why would this be so useless? It's not. We know much less about membranes, unfortunately, I think, for some reason, and they were treated as kind of passive, barriers, and that's it. But in neurons and maybe other cells, the composition and the organization plays a role. But I want to emphasize one thing, that the membranes, due to their flexibility and the heterogeneity of the composition, may not do what microtubules can, may not be able to do. They are not equipped for this reason, but also important, right, for other things. So if we figure this out, we would be able to understand better cell biology. We don't know it yet.
Totally. Totally. Yeah. Yeah, I mean, it strikes me that there's just conceptually, there's this big difference between function and communication, I mean, function especially in biology, is this incredibly holistic, all-encompassing thing that, like you say, all these channels are involved. There's chemical and electrical and piezoelectric and quantum, presumably, and when we communicate human to human, we kind of combine those. I mean, you can strip out most of the layers and just like read a book, but still the communication that's reaching you can have, hormonal and all sorts of different effects on you. You can even hallucinate, some of the channels that are missing when you're reading deeply. And so I wonder, when you think about the embodied cognition of the cell, I mean, for example, like Mike Levin's, you mentioned his experiments with planaria.
Yeah.
And he's kind of got this isolated layer. Like, what's cool about those experiments, he's kind of isolated this bioelectric layer. And then sort of, like my question earlier, like he's figured out how to talk, and say something to the layer, right? And say, make me two heads on this planaria. And then, it remembers how to do that. And so Um, you know, like Wayne had a really insightful comment in the chat after I asked that question about, how do we how do we talk to microtubules? He was like, "No, Charlie, it's the channel, not the thinker," And I hear that, but I wonder, like, that's kind of where the question is
I
like,
see.
to what extent is the structure itself a channel, and to what extent is it a thought,
It's a great question. Um, Before I try to answer, I'll say one more thing, which is, with planaria and other examples, and paramecium and all that, I think it's another example or examples of what happens in biology, that something is sufficient but not necessary, and evolution maybe finds new and improved ways of information storage and communication. So what planaria may be doing, we cannot, or salamander, right? Regrowing organs and tails and stuff like that. So it's not that it's a linear process. You may discard something and invent something better. And for this and for this reason, I think the structures become more and more intricate, perhaps. And so that's number one. In terms of, I think the deep question that you're trying or you're posing to me and others is, you know, what is the mechanism of decision making, right, in the biological system? Who is deciding? Where is that? And that is a big one. I don't know. I honestly don't know. I think. Um, Yeah, I don't know. I honestly cannot answer that question. And of course, so Albert Buehler talked about the central being the brain and the eye of the cell. Okay, he actually the eye and the brains, and of course it's microtubules. Uh, but but maybe there is some sort of, people talk about collective wisdom, right? In society, we have collective wisdom. Maybe the cell has a collective wisdom of its parts or some of its parts, right? Collectively. I don't know. I'm evading the answer, I guess.
Yeah. I mean, yeah, we sometimes, there's the idea of all of the constraints of the cell, acting on one another, in which case, if I'm following that, and I may be getting it wrong, um, there's at least a minimal set of physiological processes that have to happen in concert, for the type of knowledge to appear that is at least in the context of life, in order to
So,
remain alive. But in order to know it, at least I think it's possible that there's a more stripped down minimal version. Um,
so I want in this context, who makes the decision, what makes the decision, what I want to bring to this discussion and to think about what again. I thought about Albert Bueller's idea, and when you look at cell division, the first thing that happens when the cell decides to divide, right? We don't know how, is to divide the centrosome into two. That's the first thing that can be noticed. I don't know of anything else. And so it's a bifurcation process, right? You have a structure of two perpendicular cylinders and they float apart. What's causing this? I would say maybe some electromagnetic signal or point of decision, but by whom? The second thing is a bit more weird, but in the salamander, axolotl, I think it's a Mexican salamander or something. I had a friend, I still have Dick Gordon. He's a cell biologist from Winnipeg. He had a unique colony of these salamanders, and there is the I don't know if you know about the embryology of the salamander. It hatches an egg, and this egg can be sitting on a table for days and nothing happens. And then there's a moment, a decision, which may never happen if it's too cold, for example. But when its proper conditions are there is something called the yolk is inverted. It's a cortical rotation. The whole thing. So there's a yolk in this egg, and it's inverted or rotated from bottom to top and then splashes all over. And that's the beginning of the form being developed of this embryo. Interestingly, the force, you have to apply a torque, right? You twist it. Guess where the torque is coming from? There are cortical microtubules, which basically do some sort of paddling, like and they switch positions and they flip, and that causes this torque.
Wow.
So that is as close as I can get to answer your question. What is what is the decision-making thing? I still don't know, but I know that what is maybe the second thing that happens. And in both cases, is microtubules in different forms. In the case of this embryo, it's the egg is the microtubules aligned on the surface, on the inner surface of the eggshell. In the case of the dividing cells, is the centriolar, very intricate architecture microtubules. It's 29 microtubules.
Yeah, that's fascinating. Marco has a question.
No, thank you. And thank you, Jack. He's actually trying to use the question to explain an adding layer that with Jack we are working on, but is still too speculative. And if we stop thinking about biology, even in its original form, talking about a single cell, thinking all about all the interaction that is inside, and studying the system as isolated. The system is not isolated. Biological systems stay inside of an electromagnetic grid of which we don't know anything about. And so we are just start understanding that there is a possibility that information in biology is a continuous flow between inside and outside, and cellular membrane represent just the boundary, the interface that allow this continuous loop exchange. And we have indirect evidences, especially according to all the amount of work that has been done by Michael Levin, that in Xenopus was just able to separate timing, where patterning of information start to be related a particular function during embryogenesis. And he manipulated also the protein and the gene. And by doing this, he was able to demonstrate that there is a clear connection between structure and function. But still, where is this information coming from? And if we start thinking about system immersed inside of an electromagnetic grid and interacting with it, maybe that's a direction we could take in order to have a roadmap to understand better where actually information come in order to allow biology to do what it does, and to rearrange even in terms of evolution. And so, as Jack was just describing, we cannot take out pieces from an electrical circuit, arguing that each one is more important than the other. It's a continuum system that allows this transceiver way of interact with outside and inside patterning.
Yes, and if I may, Marco, perfect. I want to emphasize because in the beginning, I don't know if you were there, but I said I will say something about Penrose and Hameroff because this is it. This is exactly what Marco said is the fallacy. I'm sorry to say it so bluntly, but it's the fallacy of that theory because microtubules are not alive and they may have some quantum states, but these states have to be excited. They don't emerge spontaneously. So you need to provide energy for this, and you need to do this readout and read in and read out. You know what you said, Charlie. You know so that's the totality of the thing, and consciousness is not in microtubules. It's the entire system must be connected tightly to process information. I rest my case.
That's great. Yeah. Yeah, but still, even still, all this sits rather uncomfortably with the idea that all the salience in biology comes from a shared evolutionary history as a system, you know. I mean, there's you know, these could be emergent properties of parts of a system that might, you know, if what you're saying is right, might be able to be separated from the rest of the unit. I mean, when we think about novel organisms like anthrobots and xenobots that they've created and begun to test for cognitive capacities, that experiment, you know, making those xenobots with the little frog skin cells, it seems like it's explicitly separating what's supposed to be the origin of salience, which is, you know, surviving as a particular morphology is supposed to be the origin of all the salience. And it's taking that whole argument away, right? Because there's the morphology isn't even, it can't conceivably, you know, survive, you know, on its own. So,
Yeah, sorry if I may, but this has been done by manipulating cholesterol composition in membrane and then rescue. And so that's exactly an indirect because they are using the psi beta, I don't remember the acronym of the compound, but it's decreasing the cholesterol inside of the membrane. And this is consistent. So what we don't know yet is the map in molecular composition of phospholipid that create a microdomain structure that correlate to the emergence of this morphogenetic current. And once we will be able to couple structure with function, that would be it, because that would be the answer. With Jack, we are trying, I mean, we are working actively with Mike in order to find you know, an experimental setup that is going toward that direction because it's the missing layer, because he did an unbelievable amount of work understanding something that is, how to say, extremely important in biology. And in 2018 and 2022 paper, he just raised the question and he was actually arguing about the effect, possible effect of electromagnetic field and lipid membrane composition. So it's just we are just adding a layer in the mechanism that Mike was able to show. It is the layer that allow for protein to do what protein can do according to the changing module of the membrane that change according to the molecular composition in phospholipids. And so hopefully this is anyway a research path that need to be answered, even if we will find a different result from what we expect. Maybe there is no signature between a membrane composition and electrical functioning in terms of patterning.
Totally.
I have a quick comment.
Go ahead, Henry.
Yeah, so I just had the feeling that many of us just confuse the artificial created condition and the biological condition. So I'll give you an example. So in there is the Gloria Happener, which she famously say, all the cancer biologists is test what cancer can do, but not cancer does. So we actually create the new system, right? So for example, if we have a normal cell, they should, in the embryonic condition, they should go to embryonic development. So we take this away, but then we already make the machine, the information passage is displayed. But evolution, you have to think about information carrier and the system that surrounded them. So if you use artificial selection, put artificial fertilization medium, without constraint, they can go to all the different directions. In fact, like Michael's experiment, fascinating, but years ago, people use chemical trait, use different stuff. They have similar, they completely, strange behavior, everything. But that is not a new feature. Is actually the feature be constrained in the normal situation. So because have such a system, then we say, aha, we see so many things. They must mean something different. No, they did not mean something different. Just mean the genome coding the package needed in the right environment to go to right direction. Otherwise, they go to all the different direction. So understand this, then we know actually what we have done. We just take away the constraint, like Jack mentioned, the embryo development. But we know, for example, when they fertilize the egg in the frog, you do not need, change the direction. You just use the needle, touch it. The development is started, right? So the information that have potential to be displayed as a biology, but if we put it into different environment, they have different play. Don't confuse ourselves as such. So yes. I just want to say this. Yeah.
Yeah.
That's awesome. Well, guys, we're almost out of time. Without wanting to reply to Henry's comment, that's so interesting. And it's one of those, in a way, cancer cells are one of those edge cases like the novel organisms, right? That they're cells that don't necessarily have a evolutionary history, as you said, so well in your book, Henry. Anyway, I wish we had time, another 2 hours to talk about that. But I just want to thank Jack and thank everybody who's joined us today. And I'll be sure and I'll share the chat as well as the
please.
recording. And and maybe Jack, if you can, you could share your slides with me, and I could send those out as well, because that was really especially detailed and rich.
No problem. And by the way, if anybody wants to continue this conversation offline, please send me an email and I'll be happy to answer your questions, and maybe you can come up with new ones.
Totally. Absolutely. And thank you. Thank you so much for, undertaking this research and sharing it, because it's really important and fascinating.
Thank you. Thank you, everybody, for joining us and asking questions and listening patiently to this.
Fantastic. Bye. Thanks, everybody. Bye-bye.
Bye.